This is the ninth ethics seminar that's been organised by the Human Developmental Biology Initiative. Pilar has organised all of them, so we're very grateful to her for all her efforts. So the purpose of this seminar series, for those of you who haven't attended, is really to cover a whole range of issues in ethics in the broader sense that would relate to human developmental biology. And of course, sometimes these issues raise contentious subjects, which some people might have very strong views about. So we hope that we will have a fair and balanced discussion, that the environment will be sort of open, inclusive, and safe for everybody to contribute. And we really encourage active participation. There's lots of time at the end for questions and discussion, and so hopefully we'll use that time well. So I think we'll get started. Very pleased to introduce Josephine Johnston. So Josie is Associate Professor at the University of Otago in New Zealand, although she's actually in London at the moment, so this isn't the middle of the night for her. Josie trained as a lawyer. She has a master's degree in bioethics and health law, and she worked as a grant-funded bioethics researcher in North America for 20 years. During that time, she held research positions at the Hastings Center in Dalhousie University in Nova Scotia and at the University of Minnesota in the States. And she taught bioethics and health law at Columbia University in New York. So Josie works on a variety of ethical, legal, and policy issues in medicine and science, with a particular focus on the implications of emerging technologies in human reproduction, psychiatry, genetics, and neuroscience. So today her title is Perspectives on the Research Ethics Review Process, which I'm really looking forward to because this is such a hot topic at the moment, certainly in the whole area of, for example, neural organoids and so forth. So over to you, Josie. We're looking forward to your talk.
Thank you. Thank you so much, Andy and Pilar, for inviting me and introducing me and for giving me a room to work in. That's better than using my hotel room nearby. So yeah, I'm really pleased to be here and very pleased to be presenting in the same time zone as you all are, and not from, I think it's 3 a.m. right now in New Zealand. So I'll just get going here. I have several slides. It's a little bit hard to know exactly what to talk to you all about in a way, because you'll do, I think, probably pretty different things. I have had a look at what you're up to, but I'm going to share some reflections and research that I've been involved in that I think is relevant. It does go back a bit of over time a bit, so hopefully that's interesting, especially for maybe people who are younger researchers who might not have been around for some of the things that have happened. And I hope that there might be a few useful lessons in here. My own personal view is that if one thing is useful, then that's a win. So hopefully there's a little bit more than that. So yeah, I don't have any financial conflicts to share. So I just want to give a little bit of background. Like, I'm sure many of you know where New Zealand is, but I always like to put this slide up just to remind everyone. So, and I'm actually based at the University of Otago, which is in Dunedin, which is in the bottom of the South Island. So please come and say hi. Ko-wai-o, which means who am I? So this is information about me, much of which Andy covered in his kind introduction. This is my home, so beautiful. So yeah, please come visit. As Andy said, I worked for a long time in the US, and so a lot of the work that I did related to stem cell research was in the Canadian and then the US environment. So I will be drawing mainly on that work. And because I was a grant-funded researcher for like 20 years in the US, I'd had to do whatever I could get funded to do, which would be something that other people are familiar with. But I did now come to the University of Otago. And one of my jobs at the University of Otago, or my service role, is to chair the Health Research Council of New Zealand's Ethics Committee, which is the body that accredits our human subject research committees around the country. So I now have a fair amount to do with the governance of human subject research in the New Zealand environment, which is a little bit informing what I'm going to say. But most of what I'm going to talk about today relates to work that I've done in the human sort of embryonic stem cell embryo research type space. And I actually did some of this a long time ago when I was a student, and I had a contract from the government to look at some work right at the very beginning of the whole kind of embryonic stem cell research debate. They just wanted to know what other countries were doing. So New Zealand is one of these places that cares a lot about what other countries are doing and likes to learn from them. And then I had a postdoc at Dalhousie where I worked for a researcher there who was funded, or we were funded by the Canadian Stem Cell Network, which was a Canadian Institutes of Health Research group. And then probably what's really most informed my thinking about this stuff is that I served on this very busy, very active oversight committee that worked for the Weill Cornell Medical College, Memorial Sloan Kettering Cancer Center, and the Rockefeller University. And they did an unusual thing, and they shared an oversight committee. And that oversight committee looked at all embryonic stem cell research and ultimately embryo research that was done at those three institutions. And of course, Rockefeller University in particular was pretty important. So that a lot of my thinking comes out of being on an oversight committee that was focused on this area, and then some scholarship that I did along the way. So what I wanted to do today was just kind of very broadly think together or look, and I'd be so interested to hear what your experiences are and what your challenges are. But what it looks like to me is that researchers like you working in developmental biology may use materials and create models that sit at the intersection of different research oversight systems. And this is already by itself a challenging thing, to be having to deal with more than one but a situation in terms of oversight. So you might be doing research that involves human subjects. So you might be dealing with an ethics review board that looks at human subjects research, whatever that might be called in the UK setting. In the US, they're called IRBs. In New Zealand, they're called ethics committees. And then you might also be involving non-human animals in your research. So you might be dealing with whatever your institutional animal research ethics oversight committee is. And then maybe there's a bespoke committee that you are also dealing with that looks at research related to embryonic stem cells, embryos, things like that. Even with that, some research that you do might fall outside established oversight categories and also be the kind of research that attracts a lot of attention. So one of the things I've found through my service on these committees is that sometimes researchers want oversight because they know that there will be public scrutiny of their work, and it's helpful to have the backing of an ethics oversight committee and a second pair of eyes looking at things a little bit differently. So sometimes people actually came to us when we were on the escrow, the body that looked after those three institutions in New York City. They would come to us even though their research didn't fall within those boundaries, and I'll say something about that. And then I thought I'd just sort of give my view, which is that ideally, but I know not always, but ideally ethics oversight and plus scholarship and recommendations, and even just talking to people who think and deal in research ethics, this can help you navigate the various challenges that you face. And I think where you work and the kind of work you do is most likely unusually affected by these different issues related to sort of overlapping, but also sometimes holes in the research system. So I just kind of wanted to go back a little bit and sort of say how we got here. Of course, in 1990, there was 1999, sorry, there was the announcement of the derivation of embryonic stem cells from human embryos, which is anticipated by this magazine cover here. And that was a novel kind of science. And there had been some special ethics consideration around this kind of thing before the announcement of the derivation of Thompson's derivation. But after that, there was real kind of question of like, well, how does this get overseen? And in thinking about how to respond to novel science like this, I think ethics committees and the kinds of people who develop rules and laws around research ethics are always thinking about the Nuremberg Code and the history of human subject research. So that's always informing people's thinking, even if the research doesn't actually involve human beings or it doesn't involve live human beings. So something like the use of human cells or human embryos, even though they might not count as human beings in the strict sense, the history of research sort of, experimentation with humans is going to be informing how we think about responding to novel science. And that's certainly what happened, I think, in the case of the derivation of embryonic stem cells. Some of the big lessons from human subject research were applied immediately, and the main one being a focus on informed consent. And then, of course, in the internationally in this area, but also specifically in the UK, the Warnock report and research and the animal rules and laws for animals and research, those also get kind of pulled in to thinking about, well, how are we going to respond to this new kind of research? And in the US, following the decision of President George W. Bush not to put federal funding towards embryonic stem cell research, there was this kind of vacuum where, like, the research, most of it was not going to be considered human subject research because it didn't involve living individuals, and having interventions in their body or anything like that, but it involved human cells and it involved human embryos, and it was clearly controversial. And so there was a need for some kind of guidance. And so guidance was actually developed by the National Academies, which is an independent organization that has some federal funding, but is somewhat sort of arm's length, to try to give some guidance to the area. And as I said, a lot of that was informed by an existing kind of bedrock around human subject research, but also some stuff related to a history around use of embryos in research. At that time, there were no ISCA guidelines, but they later came along, not much later, and they continue to say pretty much the same thing. Sorry, I shouldn't have quite moved on then, as this guidelines here, which is that as a way to manage this kind of area, the thought was there needs to be a special type of oversight committee that looks at research with embryonic stem cells and later related research, and that would be a specialized process. So, maybe to the detriment, or maybe it's the bane of researchers' existence, but in addition to human subject research and animal research, potentially now there would be this new kind of specialized committee that would look at stem cell research. And this is still the sort of thing that the ISCA guidelines call for, that they say that there are forms of research with embryos, certain animal-human chimeras, and some other models, stem cell models, that need to be reviewed through a specialized scientific and ethics review process. So this was a way that we responded to this, these novel developments over the last 25 years. So the National Academies in the US had a series of updates of their guidelines between 2005 and 2010. And I believe ISCA's first guidelines came out in at the international level in 2006 and have just been updated just over a month ago, August 2025. And these guidelines do predominantly, I think you could summarize them as sort of saying that there are three kinds of research we don't do, and they say something about that, things that are just not permitted, research that's fine to do, and research that requires this specialized oversight. And then within that, there's a lot of emphasis on issues around informed consent. So even with that informed consent emphasis, sometimes research just doesn't quite fit. So this is an example of a situation in which there was pretty clear guidance around the need for informed consent for anyone whose cells were being used in, or embryos were being used in, stem cell research in the US. And yet it turned out that when we did a study here of looking at different stem cell lines on the NIH registry of stem cell lines that were approved by its criteria ultimately, that quite a few of them did not seem to have the information required about informed consent. So even a simple thing like informed consent from donors of cells and embryos can be quite a tricky thing to actually work through in real life. Another challenge that I've observed is that there are in this area, especially in this area of novel science, be challenges with sort of putting some meat on the bones, if you like, of guidelines that can be a bit vague. So an example of that is that the National Academies guidelines, when talking about the creation and use of non-human, so chimeric animals that have human cells in them, had asked the oversight committees to consider the consequences of the human contribution to the resulting chimera and especially whether human cells could contribute in a major way to the brain of the recipient animal and make any functional contributions to the brain of the non-human animal. So this was a piece of guidance. This was basically the guidance that the National Academies gave for oversight committees looking at non-human animal chimeric research. And as a member of an oversight committee, I just found this very, very difficult to actually interpret and to use. And we actually got some help from scientists to try to understand what this might mean. Honestly, at the time, the ISCAR guidelines were also not that much more helpful, a little bit different. So we did a project to try to give some kind of depth to this question of, well, what is it that we're really worried about when we're looking at this area? So I just kind of wanted to include this to kind of share with you that, like, sometimes even the oversight committees, when you're really dealing with novel research, are being asked by their guidelines to do things that are quite vague and sometimes might require help. So I think our researchers who came to our committee sometimes felt like, why are you asking me questions about whether or not what way we think the brain of the non-human animal might be, given that it has human neurons? And it's because we were trying to implement, use this guideline to help us understand whether to approve the research, but we were also a bit lost. So there's also just like this, I think, is also a common element to when you're working in this kind of novel area that you just have. Sometimes it's just really hard to know how to follow through on guidelines. And actually, in response, we did a project on this issue, like what actually do we think is really going on in chimeric research, what's important. And we kind of concluded that actually animal welfare was the main issue for this area, not so much some of the more sort of science fiction issues around the mixing of humans and animals, and that there was a need for some interplay between the government oversight that was provided by specialized oversight committees like embryonic stem cell research oversight committees, but also the animal research oversight committees. So there needed to be a kind of breakdown of the silo there to try to get some back and forth so that the committees could be sure that they were looking at the right things. Okay, so another issue I just wanted to touch on is that sometimes there can be challenges with old rules in this area. And I think one of the best examples of this is the 14-day rule, which obviously is a major topic right now, or in the last few years. And of course, this is a paper that I published with Insu and another colleague. And we were looking at where this rule came from, and this was all in light of the fact that there was some new science coming out of Rockefeller University that was pushing the boundaries of the 14-day rule. I don't know if it's good news for a UK audience, but actually this earliest reference I found to something like the 14-day rule was actually in a US publication from 1979. Where it surveyed a variety of different possibilities for what kinds of limits you could put around this research. But of course, it's very familiar to people in the UK because of the Warnock report really kind of did sort of solidify around this. And then the US picked that up in a more specific, definite way. But then suddenly there was a whole lot of research that was pushing this boundary. And one of the things that we sort of found when we looked at this is that the 14-day rule itself wasn't really intended to be like the be all and end all of some sort of moral line between what research you can do and research you can't, but it was a public policy tool that was trying to carve out a space for scientific inquiry, but also show the public that scientists understand that this is a controversial area and that people have different views on it, and they're not without limits what they're going to do. And so I think that's a really important lesson to take generally for this area, that like a lot of what we're doing when we make rules and do oversight in this area is not finding a moral truth necessarily, but trying to balance the need for the research with the need for the public, with all of its different views, to see that there are some guardrails around science. So yeah, and one of the very important components of that is being really clear about why the research is worth doing. Okay. So like I said, I think the supporting research is really important, but also this is a very vague sentence, accommodating diverse moral concerns, but it can then translate into a very specific rule around, for instance, how long you're going to culture embryos in vitro. Some people argue that if you're going to change a rule like this, I think changing a rule might be necessary, but it's actually quite difficult to do, not just because like sort of for pragmatic reasons, but because you if you change a rule, you have to also deal with the fact that it looks like you're moving the line just to accommodate science, like your line was not really a real line in the first place. And so some people have said that they think that one way to kind of work through this is to do public engagement and to really get engaged with the public and sort of open out science a bit more than find out what people want. So not just what oversight committees want or scientists, but also what public concerns are. This is a very difficult, I think, thing to do. It's a very difficult thing to do in a large country, and it's a very difficult thing to do today. And I just wanted to kind of complete this issue around the 14-day rod by pointing out that it looks like ISCAR has no longer got any specific boundaries around time for embryo culture. Okay, so if you'll indulge me, I just wanted to offer like a few little bits of advice that I have come up with based on different conversations I've had over time with researchers working in generally in, I think, the field that you're in. One of them is that I recommend that I think it's really important to explain the research that you do and why it's needed. So to connect it to real world questions, and including just the question of like trying to understand life. Basic science it's got its own set of reasons. But when you do, I think it's important to just check that you are using plain language and that you're not unwittingly saying something that gets you in even more hot water. And I had a recent example, I won't say where it's from, but where I was consulted before something was published, and the research was justified with the following statement: fertility rates are decreasing around the world, which together with increased maternal age creates a rapidly rising demand for effective IVF. So the heart of this is good, but what the researchers, what I was kind of helping the researchers do was to sort of understand why that kind of statement can also be a bit damaging because of contemporary political and moral issues around pronatalism and sort of whether it's mothers that are the women sort of age at first birth that's the problem. So we rephrase this to make it just a little bit less like blamey. Another example, a recommendation I have is to use plain English. This is I think sort of weirdly topical, another paper from Untitled Chapter that came out quite a long time ago, but they called this new thing that they had figured out, they called it 3-parent IVF, which I think maybe they thought was cool language, but I felt like it really obscured what was going on and was quite inflammatory. So if you've invented a new process or a new thing and you're thinking about giving it a new name, I just think it's really good to be quite plain and descriptive, and generally speaking, not great to kind of come up with some cool new name. 3-parent IVF for mitochondrial replacement suggests that suddenly this thing has 3 legal parents, and you can imagine how that got picked up. Another recommendation is actually then to just be really precise and accessible. So in our Chimera project, we were trying to get away from saying that these things are chimeras, because that's a monster. And also actually we found the word humanization was used quite a bit, and that's also quite ethically laden to a non-expert ear. And sometimes you could just say something like you created a chimeric mouse or a mouse with human cells, or instead of using phrases like a chimera or that you created sometimes a humanized mouse. Just being really clear and precise and not quite so flashy is quite helpful. I think it's also really good for people in your field to sometimes say what you're not doing in your papers even. So something like this model is not a human embryo and it cannot become a human embryo because it's missing X and Y. I think you can assume that some of the people reading the work that you do will not be scientists, they'll be journalists or others, and they will be reading the beginning and the end, especially to try to understand what's being created, and it's good to spell it out. And if it's something that can or can't do something, like if instead of saying if you're like a lung organoid or something, maybe also explain like why it's not actually a human lung completely, or what it can or can't do, so that people can really see the boundaries of what you're doing and and some of the limits of it. And then I guess the other thing I would just say is that working in the area that you work in, I think there is also no pleasing everybody, and there will be some controversy. And so this paper was published yesterday. Induction of experimental cell division to generate cells with reduced chromosome ploidy is, oh, sorry, that's the wrong paper. No, that's the paper, yep. And that's being reported as human embryo made with skin may one day produce 2 dad babies. So there's just sometimes no way around it. So thanks. Those are just some reflections and some thoughts and research, and I'd be really happy to follow up on any of it in the question and answer. So thank you.
Thank you, Josie. That was great.